Well students, in the previous class we have discussed one of the important cell organelle, that is endoplasmic reticulum. In this class we will start with another organelle called as Golgi complex. So this Golgi complex has many names, like Golgi complex can also be called as Golgi apparatus or Golgi bodies, or also called as dictyosomes. So these are the several names for Golgi complex. You can just remember Golgi complex or Golgi apparatus. And you know, Golgi complex is one of the important cell organelle present in the cytoplasm of eukaryotic cell. Actually, it is situated very near to nucleus. When you draw a cell, for example, plant or animal cell, in the animal cell, in the center, so this is the nucleus. Very near to nucleus, you can observe some concentrically arranged structure called as Golgi complex. So this is one of the important cell organelle present in the cytoplasm of eukaryotic cell. But there are some exceptions where the Golgi complex is absent. For example, in mammalian RBC, we cannot find the presence of Golgi complex. So that we can say that Golgi complex is absent in the mammalian RBC, and even they are absent in the CO2 elements. The CO2 elements of phloem. When you study the structure of phloem, you will come to know the CO tubes. Especially in the CO tubes, the Golgi complex is again absent, and even they are absent in the sperm cells of bryophytes and pteridophytes. In the plant kingdom, there are several divisions like algae, bryophyta, pteridophyta, etc. So in the sperm cells of bryophytes and pteridophytes, the Golgi complex is absent. So generally they are present in the cytoplasm of eukaryotic cell, but they are absent in mammalian RBC, the CO2 elements of the phloem, and even the sperm cells of bryophytes and pteridophytes. So this Golgi complex, it was first discovered by an Italian scientist called as Camillo Golgi. So Golgi complex was first discovered by an Italian scientist called as Camillo Golgi in the year 1898 in the cells of barn owl. So there is a species of owl called as barn owl. So especially in the cells of barn owl, he discovered the presence of Golgi complex. So who discovered Golgi complex? It was discovered by Camillo Golgi in the year 1898 in the cells of barn owl. Later, the structure of Golgi complex is observed under the electron microscope by another scientist called as Dalton. So the structure of Golgi complex observed under the electron microscope by Dalton. Hence, Golgi complex can also be called as Dalton complex. Golgi complex can also be called as Dalton complex, as the structure under the electron microscope was studied by Dalton. Then, what is the size of the Golgi complex? Actually, the size varies according to the function. If for example, if the function is a secretory function, if some cells performing secretory function, in such a secretory cells, the Golgi complex is usually bigger. If for example, if this cell performing secretory function, in this cell, the Golgi complex can be bigger. And we can give the examples for secretory cells like liver cells and pancreatic cell. In those liver and pancreatic cells, the secretions are more. That's why the Golgi complex is usually bigger. But Golgi complex is smaller in case of non-secretory cells like muscle cells. So as they are non-secretory in function, then the Golgi complex is usually smaller. But generally, a Golgi complex usually measures about 2 microns in diameter and 0.5 microns in width. So generally, the Golgi complex measures about 2 microns in diameter and 0.5 microns width, but it may be bigger or smaller according to its function. Then, the number of Golgi complex per cell. So when you draw the animal and plant cell, you have shown one single Golgi very near to the nucleus, but we cannot say that always it is one. Especially in case of animal cell, we can expect only one Golgi complex per cell. In every animal cell, there is one Golgi complex in the animal cell. But it may be 6 to 12 in case of plant cell. It may be 6 to 12. Actually, you have drawn only one Golgi complex in the plant cell, but it may be 6 to 12. And especially in case of Chiara, it is actually a green algae called as a Chiara. In this Chiara, the rhizoidal cells, they may contain up to hundreds of Golgi complex. So especially it is a green algae called as Chiara. In this green algae, the rhizoidal cells containing about hundreds of Golgi complex per cell. What I am saying? In case of animal cell, only one Golgi complex per cell, but in plant cell, 6 to 12. But the rhizoidal cells of Chiara may contain few hundreds of Golgi complex. Then, when we come to the structure of Golgi complex, actually the structure of Golgi complex is mainly made up of three components, namely cisternae, tubules, and vesicles. So, cisternae, tubules and vesicles, these terms you already heard when we study the endoplasmic reticulum. Even the structure of endoplasmic reticulum comprising of the cisternae, tubules and vesicles. Even also the structure is quite similar to endoplasmic reticulum. See, look at the diagram. So, this is the endoplasmic reticulum, mainly made up of three components. These are the cisternae. Here in the diagram we cannot find the presence of tubules, but there are vesicles. So what is this cisternae? Actually, cisternae, these are many disc-like or sac-like structures, and usually they are flat. The many flat disc-like or sac-like structures which are arranged in parallel bundles. See, these are the disc-like structures which are arranged in parallel bundles. And one Golgi complex may comprise of 4 to 8 cisternae. The number of cisternae varies from 4 to 8. Here we can look at here, each one is called as a cisternae. Here we can find the 4 cisternae. Usually in a Golgi complex, the cisternae varies from 4 to 8. And they are always arranged in parallel bundles, and each disc-like structure or each cisternae, it is a disc-like structure with an expanded margin. This expanded margin is called as Golgi vacuole. The expanded margin is called as a Golgi vacuole. Try to understand. Cisternae, these are many flattened disc-like structures which are arranged in parallel bundles, and each disc-like structure having an expanded margin called as a Golgi vacuole. You will come to know the importance of Golgi vacuole later. And these cisternae, actually what you can see, the 4 to 8 cisternae, they are arranged concentrically near the nucleus, or we can say they are very close to nucleus or endoplasmic reticulum. What the endoplasmic reticulum you can see. So this cisternae of Golgi complex, they are very closer to the nucleus or endoplasmic reticulum. The reason also you will come to know later. And as they are concentrically arranged to the nucleus, arranged very close to nucleus with a distinct polarity. Polarity in the sense they have two ends. One is called as convex end, the other one is called as concave end. See, in this, actually the Golgi complex is arranged like this with distinct polarity. This surface is called as convex surface or convex region, and this region is called as concave. So this is called as a convex region and this is called as a concave region. And this convex region, what you can see, the bulge-like structure. So this convex region is called as cis phase or forming phase. And the opposite one, the concave region, is called as trans phase or maturing phase. I will tell you why it is called forming maturing phase. We just remember the system name which are arranged very closer to nucleus with a distinct polarity, a convex region and a concave region. The convex region is called as cis phase or forming phase, and the concave region is called as trans phase or maturing phase. So that is about the system name. Then what are these tubules? Actually, these tubules are the network of tubular structures which are actually arising from the peripheral region of the system name towards the maturing phase. So these are the network of tubular structures which are usually arising from the peripheral part of the system name, especially towards the maturing phase. And these vesicles, the vesicles are usually a small circular sac-like structures. Again, they are obtained from or arising from the tubules towards the maturing phase. The tubules also towards the maturing phase and vesicles also towards the maturing phase. The tubules arising from the peripheral part of the system and vesicles arising from the peripheral part of the tubules. And we can find some larger vesicles. Actually, these larger vesicles are called as vacuoles. So, in a cell, how the vacuoles will be obtained means they are obtained from the Golgi complex. So, how they will be obtained, that we will come to know when we study the functions of Golgi complex. So, the larger vesicles very near to the Golgi complex are called as vacuoles. So, this is about the structure of Golgi complex. But here what is very important means the functions of Golgi complex are very important. As I said, the functions of endoplasmic reticulum is important. Even the functions of Golgi complex is also very important. Now we will discuss the functions of Golgi complex. So the main function, the principal function of Golgi complex is that packaging of materials. Packaging of materials. The principal or the first and foremost function of Golgi complex is the packaging of materials. So what is actually this packaging of materials? See, for example, here we can find the presence of nucleus and there is an endoplasmic reticulum. On the endoplasmic reticulum you can find the presence of ribosomes, and the main function of ribosomes is the protein synthesis. If these are the vesicles of endoplasmic reticulum, see, vesicles can be seen in the endoplasmic reticulum also and even in the Golgi complex also. So the vesicles from endoplasmic reticulum which contain ribosomes, and these ribosomes help in synthesis of proteins. So all those proteins are these vesicles. They fuses with the Golgi complex. Actually, what the proteins that are synthesized by ribosomes, they cannot be directly released into the cytoplasm or cannot be directly given to other cell organelles. First, they will be modified in the Golgi complex. So, what the vesicles which are present, they first fuses at the cis phase of the Golgi complex. Later, these proteins that are synthesized by ribosomes, they will be moving with this system name and they are modified. Later they will be packaged in these Golgi vesicles. So after packaging, so this portion, the expanded region may be pinched off to form either vacuoles or lysosomes. So that is what I am saying. The principal function of Golgi complex is the packaging of materials. So the packed materials that will be discharged are delivered to the target cell organelles. So whatever the cell organelles that require proteins etc., all those things will be modified and packaged within the Golgi complex. That is the first function. And the packaged materials are in the form of vesicles, especially the vesicles of endoplasmic reticulum. They fuses with the Golgi complex, especially at the cis phase, and they are moving towards the maturing phase. So here they are forming, later here it is maturing. That's why we have given the names forming phase and maturing phase. The vesicles from endoplasmic reticulum fuses at the cis phase and moving towards the maturing phase. And now you can understand why Golgi complex is situated very closer to nucleus and endoplasmic reticulum. So here the vesicles of endoplasmic That's why these two are very close to each other. Then, as I earlier mentioned, the vesicles of endoplasmic reticulum, if they contain ribosomes, they are called as rough endoplasmic reticulum. So, this rough endoplasmic reticulum having ribosomes, and ribosomes are synthesizing proteins. So, before the proteins are discharged, they have to be modified within the Golgi complex. So that the vesicles containing ribosomes, their fusers at the cis region, they will be releasing the proteins into the cisternae. Now these protein molecules are moving along the cisternae. Later within the cisternae they are modified. So they are moving all over the cisternae and they are modified and packaged at the Golgi vacuole. And this Golgi complex is the important site for the synthesis of glycoproteins and glycolipids. If you can remember the fluid mosaic model of plasma membrane on the lipid molecule. So in the lipid bilayer there are lipid molecules along with the proteins. As you know, intrinsic and extrinsic proteins. Here some carbohydrates are attached to lipids and some carbohydrates are attached to proteins. So they are called as glycoproteins and glycolipids. So this Golgi complex actually helps in attachment of carbohydrates to the proteins and lipids so that it can able to form the glycoproteins and glycolipids. So you should remember the Golgi complex, it is one of the important site for the synthesis of glycoproteins and glycolipids. It means helps in attachment of carbohydrates to proteins and lipids. And Golgi complex, it is the site for the site for the synthesis of very important enzymes and hormones. So several hormones and enzymes can be synthesized from Golgi complex. And another important function is the acrosome. Actually, when you study about the sperm, the main gamete is called as a sperm. Actually, the sperm region having a head region and tail region. At the head region, there is a cap-like structure called as acrosome. So in the sperm, you can take the human sperm. In the human sperm, there is a cap-like structure called as acrosome. Actually, that acrosome is nothing but the modified Golgi complex. So this Golgi complex only, which is present in this sperm, it is modified to form acrosome. So this is very important function that you will study in your second PUC also. So acrosome of sperm is called as modified Golgi complex. And this Golgi complex helps in synthesis of various vacuoles and lysosomes. So Golgi complex helps in formation of lysosomes and vacuoles. Even it helps in formation of retinal pigments. So when you study about the eye structure, you will come to know about the retina. So Golgi complex helps in formation of retinal pigments. It helps in formation of cell plate during cell division. So during the cell division, what the cell plate is formed like fragments, fragmoflage. So these cell fragments, what you can see, they are actually the fragments of Golgi complex and endoplasmic reticulum. The fragments of endoplasmic reticulum and Golgi complex, they are arranged like this to form the cell plate during cell division. And Golgi complex, it is also helps in formation of middle lamella. So after the cell plate formation, there will be accumulation of calcium and magnesium peptide to form middle lamella. If you can remember, middle lamella, it is associated with or accumulated with calcium and magnesium peptide. So that accumulation of peptide substances is mainly because of Golgi complex. So the helps in formation of cell plate and even it helps in formation of middle lamella. And another important function, the root cap cells. When you take the root, here there is a root cap. So these root cap cells are rich in Golgi complex. So the Golgi complex which are present in the root cap, they help in secreting some fluids. It will provide lubrication. So actually it will secrete the mucilage. So this mucilage will provide lubrication. Lubrication is nothing but to avoid the friction. It is like a grease or oily like substance. It will avoid the friction. Likewise, the Golgi complex, which are rich in root cap cells, they will secrete mucilage to provide lubrication. So these are the various functions of Golgi complex. But organelles are intracellular targets. And second function, the packaged materials are in the form of vesicles. The vesicles of endoplasmic reticulum fuses at the cis phase and moving towards the trans phase. The ribosomes, they will be synthesizing several proteins, and all those proteins are modified within the Golgi complex before they release. So how they can be modified? They fuses at the cis phase, moving along the cis domain and modified. Later they will be discharged into the cytoplasm. And it is an important site for synthesis of glycoproteins and glycolipids. Also helps in formation of several enzymes and hormones, formation of lysosomes, retinal pigments, cell plate, middle lamella, and even the acrosome of sperm is called as modified Golgi complex, and root capsules are rich in Golgi complex, helps to secrete mucilage that provides a lubrication for the root. So this completes the Golgi complex. Here we have discussed who discovered Golgi complex and the structure observed by Danton, and why it is called Danton complex, everything you know. Here we have discussed the size of the Golgi complex, number of Golgi complex, structure, and very importantly functions. So this completes about the Golgi complex. Now we will discuss few objective questions related to Golgi complex. See the first question. Cis and trans phase of Golgi body are dash and dash respectively. Option A convex and concave, option B concave convex, option C convex convex, option D concave concave. So here cis phase is convex and trans phase is concave. So option A is correct. Second question. Which phase of Golgi apparatus receives the materials packaged in the form of vesicles from endoplasmic reticulum? So here option A is cis, option B trans, option C both A and B, option D none. So actually the cis phase is receiving the vesicles from endoplasmic reticulum. So option A is correct. Next third question. Golgi apparatus is an important site for the formation of option A proteins and lipids, option B glycoproteins and glycolipids, option C carbohydrates and proteins, option D glucose and lipids. You know that it is an important site for formation of glycoproteins and glycolipids. So option B is correct. Next fourth question. Which structure is formed by the process of packaging in Golgi apparatus? Option A ribosomes, option B protein granules, option C lysosomes, option D centrosomes. Here correct answer is lysosomes, because the vacuoles and lysosomes, they are formed from the Golgi complex because of the packaging of materials. I told you these are considered as the part of the endomembrane system as they are closely associated, or their functions are coordinated with one another. See, when you look at the functions of endoplasmic reticulum also helps in formation of lysosomes and vacuoles. Even Golgi complex also helps in formation of lysosomes and vacuoles. So their functions are coordinated. Hence, we consider Golgi complex, endoplasmic reticulum, all these as a part of endomembrane system. So this completes about the Golgi complex. In the next class, we will take up the